Hello students, I hope you are doing well. I am Honey Rachel and I teach Biotechnology in K.B.N college. Welcome to this session. In this session, we'll try to learn something interesting. So before I start, I want to ask a question to all of you. Have you heard of these names, sickle cell anemia and cystic fibrosis, Down syndrome, Turner syndrome? You might have heard these. What do you think is common in all these names? Cystic fibrosis, sickle cell anemia, Down syndrome, Turner syndrome. You have guessed it right. All these are diseases which are caused due to mutations. And where and how are these mutations caused is the second question. You have guessed it right. In the genetic information, the mutation has occurred, and because of that mutation, we have got all these diseases. If you know, we are exposed to different carcinogenic agents in our life. We are exposed to different environments which might contain some carcinogenic agents like radiation, UV light, chemical carcinogens. All these cause damage to the DNA. Not only the chemical carcinogens, but whenever the DNA gets replicated, it will undergo some changes which are not usual and which are damaging to the DNA. But you know the special thing about our body? Whatever gets damaged, it gets repaired when it's given time. Yes, after we are exposed to chemicals, radiations, our body tries to repair that damage. Even at genetic level, our DNA, which has got damaged during replication, also get repair regularly. That's why even with those damaging effects, we are surviving without any genetic manipulation. But in some cases, the mutation or the damage that is done to the DNA, it stays like that without being repaired, and that condition of non-repaired DNA, we call it mutation. And in that cases of mutation, the damage is sometimes visible, and it's sometimes damaging diseases like sickle cell anemia. And the mutations, they can be categorized into two. Mutations that happen at genetic level, that is in the sequence of genes, and the other is mutation that happens at chromosomal level. As you can see in the pictures, this is genetic level mutation, that is genetic mutation, and this is chromosome mutation. Two are quite similar but are very different. Let us understand more about what's genetic mutation and what's chromosome mutation, shall we? Let us start with chromosome mutation first. Chromosome mutation, as its name says, the mutation happens, or the change or damage happens at or in chromosome. You all know what chromosome is. The condensed form of genetic information, DNA, is called chromosome, and we can observe these chromosomes during which phase? Yes, metaphase. And during the cell division, during metaphase, if there are any damages, if there are any problems, that will affect the chromosome, and then we call it chromosome mutation. There are 4 types of chromosomal mutations and they are listed here. Let us start from the first one, duplication. Sometimes what happens, fragments or the genes of a chromosome, they appear more than once in the chromosome because of some mutation. We call that duplication. If you observe this picture clearly, this is the original chromosome which has A, B, C, D, E, F genes. And after mutation, what happened is it has 2 B's. B's in double, right? We call this condition as duplication and this mutation as duplication chromosome mutation. The second one is deletion. Many times due to mutagens around us, few fragments of the chromosomes are cut and removed unnecessarily. As you can see in the first frame or first picture, we have 6 genes and later we have only 5. The B is missing, right? That's because of deletion mutation. Third is inversion. Let us see what that is. Inversion, we can say another term along with inversion, that is switching of places. That is, if you can observe the first chromosome, it has A, B, C, D, E, F in a sequence in a row. And after mutation, what we have is A, E, D, C, B, F. What happened here is E, D has switched their places and C, B switched their places in inverse. This happens sometimes and the map completely gets changed because of inversion, the map of genes. Let us understand the last one, translocation. In translocation, the genes or fragments of chromosome, they pinch off and they get attached to the other chromosome. Observe here. We have 2 chromosomes, one with capital letters and one with small letters. And after mutation, you can see the capital letter one with last 3 genes of small letters. This fragment is actually the fragment of other chromosome. And this fragment which has EF, it shifted to the another one. This is the change in location completely to the other chromosome. This also happens during replication. So these are the chromosomal mutations which happens in the chromosomes: duplication, deletion, inversion and translocation. Let us understand the other type of mutation. Before that, if you observe this picture carefully, this kid has Down syndrome, and the Down syndrome is because of an extra chromosome, and that is one of the example of chromosomal mutation. Not only Down syndrome, Turner syndrome is also an example of chromosomal mutation. And what about genetic mutation? This mutation is at the level of genes. We all know we have 4 nucleotides A, T, G, C which make up the genetic material. When there is change in that material, even one nucleotide change can cause a drastic effect, can cause a drastic change. How? Let us understand. So the first is missense mutation. This frame is a DNA, and from the DNA mRNA is made, and from mRNA protein is made, we all know. When the DNA code has change, has mutation, at one nucleotide, we call it missense mutation. Observe this. Here we have CAD, which codes for histone protein, and here we have G instead of A, and we got ARG, arginine. The complete amino acid has changed, and change in amino acid will leads to change in complete protein, right? So this missense mutation is caused because of only one nucleotide. We call it point mutation. The mutation that happens at only one point at one nucleotide. A good example to this missense mutation is sickle cell anemia. Only one nucleotide change causes difference in the hemoglobin protein, which causes difference in the shape of the red blood cells, and because of that the patient with that genetic mutation would experience a very severe effects, like he wouldn't be able to absorb good amount of oxygen, swollen legs and swollen hands, and also enlarged spleen and liver is the condition. Let us go for the second type, nonsense mutation. Sometimes that one particular change in amino acid will cause such a damage, like in this case. Look at the first frame, we have mRNA and also protein. In mRNA we have nucleotides. Every 3 nucleotide is a codon, and each codon codes for one amino acid. If one nucleotide is changed, the complete codon is changed and the amino acid is also changed. Consider this, we have AAG, which code for LYS amino acid. If instead of A the mRNA is mutated with U, one change in nucleotide. Now what it has changed into? AAG has changed into UAG, and you know UAG is a stop codon. Whenever there is stop codon, the protein synthesis stops, and that happens here as well. Whenever the A is mutated with U, the protein synthesis suddenly ceases. When there is no complete protein, then there is no functional one, and then there is problem. And this case we can observe that in cystic fibrosis, the nonsense mutation, the same case is here. The cystic fibrosis, what happens is a particular protein its synthesis is ceased because of mutation, and because of that the body will not be able to synthesize that protein, and because of that the body of the patient, especially at the tracts, respiratory and the intestinal tracts, are filled with the mucosal layers, and that causes a lot of discomfort, and that is cystic fibrosis, that which is caused due to nonsense mutation. Another type is silent mutation. As the name suggests, it keeps quiet and doesn't cause any damage to the body. And how does this work? Whenever the mutation is caused in the genetic information and that mutation doesn't change the amino acid, then we call it silent, for example. Both the codons GAA and GAG, they both code for the same amino acid GLU. And whenever there is change in nucleic acid, whenever the A is replaced by G in GAG, we get GLU and there is no change at all. The silent mutations does happen, and these are not expressed like other diseases like sickle cell anemia. We can only observe these if we do the gene sequencing. Otherwise, they are silent. The last one, the last mutation of genetic mutation is frame shift. Whenever there is addition or deletion of one or two nucleotides, this happens. There is no interchange, but there is complete addition or deletion. How? Observe the second frame where C and G are removed from the original sequence. If you see the first sequence, we have the sequence of amino acids: alanine, threonine, glutamate, leucine, arginine, serine. But from the CG are removed, and we have only GAG. That's why what our body does is it prepares an amino acid, it adds an amino acid which the code is made of GAG, so glutamate. But the structure of protein is being changed, and because of that the damage happens. If you observe carefully, there is a shift in the frame of codons during addition and deletion, and we call this frame shift mutation. This case we can observe in Tay-Sachs disease. Because of frame shift mutation, one particular protein, it gets damaged and becomes improper function. It does improper function. Because of that, there is swelling in neurons, and there is red spot in the eye, and it causes narrowing of blood vessels. Because of that, the child would experience a lot of differences like less cognitive development, and less motor development, delayed motor actions. This is all because of frame shift mutation. And these are all the different kinds of mutations. As you have observed, we have two types, that is chromosomal mutation and the other is genetic mutation. I hope you learned something interesting and enjoyed this session. Thank you for joining us.